Good afternoon, everyone. My name is Irene Cobo. I'm a postdoc at the Department of Ecology and Evolutionary Biology at the University of Connecticut. And I'm going to talk about CartograPlant, which has been the main topic of my two-year postdoc at UConn. So CartograPlant is a web-based application that integrates, visualizes, and analyzes genotypic, phenotypic, and environmental data from georeferenced plants, including forest trees. And this web-based application is relevant and timely nowadays because, as we all know, climate change is threatening plant health and productivity at all spatial scales. However, it remains largely unknown whether plant breeding can keep pace with the rate and direction of this environmental change. On the other hand, the incidence of invasive pests and pathogens is increasing due to globalization, and this tendency is being exacerbated due to current climate change. Another specific threat for forest trees is the illegal logging and deforestation, and it is also worth mentioning that we are in a post-COVID era, so healthy and productive terrestrial ecosystems, including forests, must be maintained in order to reduce the risk of and also improve their responsiveness to future pandemics. As a result, the match between genotypes, phenotypes, and new environments is key in order to identify candidate genes of resilience to these biotic and abiotic stresses. On the other hand, the illegal logging and deforestation can be tackled by identifying the timber origin using chemical, genetic, and anatomic tree data. Thus, we can conclude that tools that collect, integrate, and facilitate these genotypic, phenotypic, and environmental data, such as CartograPlant, are critical nowadays. Regarding the data integration, because as I have mentioned, CartograPlant integrates, visualizes, and analyzes data, there are three main types of raw data available in CartograPlant: genotypic, phenotypic, and environmental. However, the question is, how can these disparate data types coming from different studies be integrated? And the answer is thanks to the metadata collection using ontologies and standards. Regarding this raw data and the associated metadata is available in CartograPlant thanks to two main sources. One of these sources is the direct submission of studies by users and also by our biocuration team. Through the triple PlantPop GenSubmit or TPPS pipeline, it is worth mentioning that this pipeline hosts any study containing any combination of genotypic, phenotypic, and environmental data. And once the study is submitted, it provides a DOI to the user. The main objective is to ensure the fairness of the data, that is, its findability, accessibility, interoperability, and reusability. The other main source of data in CartograPlant are the biocuration effort of our affiliated databases. As a result of these two main sources of data in CartograPlant, CartograPlant currently hosts about 8 million of plants belonging to 500 species, 200 genera, 43 countries, 200 studies. And as a result, we have about 58 million of genotypes, 1.2 million of phenotypes, and 931 environmental layers in CartograPlant. Regarding the data visualization, because again, CartograPlant integrates, visualizes, and analyzes data, the CartograPlant interface is divided into two main panels. The left panel, which is the action panel, where users can interact with the map and the plants, and it is located on the left part of the screen. And the right panel, which is a worldwide interactive map that shows the selected plants and the plants and the environmental layers selected on the left panel. For instance, regarding the environmental layer visualization, it can be performed by clicking this environmental layer button here, this tab. And by doing so, all the environmental layers available in CartograPlant are displayed. So for instance, by clicking on one of them, the environmental layer of interest is displayed in the right panel in the worldwide map. And also this worldwide map is, as I have mentioned, interactive. So by clicking directly on the layer on the map, information about the environmental layer at this specific point is displayed, as you can see. The same can be done with the plants in the worldwide map. So by clicking directly on a plant in the worldwide map, information about this plant is displayed, including the more detailed information about the study to which this plant belongs that can be displayed by clicking this study info button here. And also the plant can be selected for further data analysis by clicking this green button here. Another way to select plants for further data analysis can be done by two different ways, basically. One way is by using the left panel, by clicking at this filter tab here that allows users to filter the plants by different criteria, including taxonomical information, molecular markers, plant structure, phenotypic attribute, or study title, author, and accession. Another way to select plants for data analysis is by drawing a polygon directly on the map by using this polygon button here. And once the plants of interest are selected, authenticated users can perform data analysis by clicking this Analyze button here. Once users click this button, this window is displayed that says CartograPlant Analysis. And as you can see, there are several tabs there. So the first tab that says Begin basically provides a unique analysis ID, in this case, the number 77. Also allows users to give a name to the analysis, and also the users are strongly encouraged to create a workspace by clicking this button here. Now, this workspace allows to store the data and the results for the data analysis for further analysis, for analysis that can be performed in the future. Once the users have created the workspace, the following tabs are mostly focused on data filtering. So again, the selected plants contain phenotypic, genotypic, and environmental data. And these tabs provide tools to the users in order to filter this data, the most interesting data for further data analysis based on the biological questions the users may want to ask. So for instance, regarding the phenotypic data, CartograPlant provides the studies, the number of studies to which these plants belong, in this case, 4 studies, also the number of traits detected, and also provides the overlapping status of the detected traits. So for instance, some of the traits overlap among all the selected plants, in this case, 10 plants, but others overlap only among 2 of the selected plants. So based on this overlapping status, the users may select the most interested traits for their analysis. Once the users select the traits of interest, a histogram is displayed showing the phenotypic distribution of the selected traits. This histogram is interactive, so the users, by moving these 2 red bars here, can select the most interesting traits based on their distribution. So for instance, depending on the questions they want to ask, they can be interested on the central traits of the distribution or the tails of the distribution. The same can be done with the genotypic data. So in this case, again, the number of studies to which the plant belongs are displayed, the selected plant belong are displayed, and the genotypes, the overlapping status of the genotypes is displayed by means of a Venn diagram, making use of in this case, of the 4 studies to which the plant belongs, the selected plant belong, and again, this Venn diagram is interactive. So the users, by clicking on the different areas of the Venn diagram, can select the genotypic data of interest based on their overlapping stages. In addition, users can also select the SNPs and the individuals by quality. So different quality filtering criteria are available. They are mostly the standard quality filtering criteria mostly used in the majority of the genome-wide association, large-scale genomics, and population genomic studies. So once the user select the filtering criteria of interest, again, the data is displayed in a interactive histogram so that the users can select the most interesting SNPs or individuals based on their quality. And finally, the environmental layers of interest can be selected in this tab here that displays all the environmental layers available in CartograPlant. And it is worth mentioning that all the environmental layers available in CartograPlant are suitable for all the studies available in CartograPlant because all the plants available in CartograPlant are georeferenced. And the last tab allows users to perform data analysis. So there are several analytical workflows available in CartograPlant that I'm going to explain in more detail in further slides. But before that, it is worth mentioning that this data analysis is possible thanks to a Galaxy instance. So for those that don't know what Galaxy is basically a web-based program that allows users to perform bioinformatic analysis of high-throughput data without the need of coding skills because it is very user-friendly. And finally, these are the 7 analytical workflows currently available in CartograPlant. It is worth mentioning that they are mostly focused on SNPs because they are the most popular molecular markers in biological studies. So the first analytical workflow available is SNP quality filtering again. I know that it is possible to perform this quality filtering in previous tabs in CartograPlant analysis, but sometimes the visuals provided by CartograPlant are not enough in order to select the best thresholds for your data, because it can be tricky. So in order to handle this potential issue, we provide a SNP quality filtering workflow that makes use of a program called Link Imputar. So this program, what it basically does is to perform different combinations of thresholds for the data, and provides the number of retained SNPs and the accuracy of the data after the different combinations, so that the users can select the best combinations of thresholds for their data. Once the quality filtering is performed, Link Imputar also performs the imputation of the lacking SNPs, which is very powerful because this imputation enhances the association power of further analysis. In addition, CartograPlant also offers a SNP remapping workflow that allows to remap the SNPs against other versions of the reference genome. This is very useful for certain analysis, such as meta-analysis, that requires that the SNPs are mapped against the same version of the reference genome, the SNPs belonging to different studies. But also it is very useful because it allows to have the SNPs of the studies available in CartograPlant the most updated as possible, without the need of perform resequencing of the data, which is very expensive and time consuming. In addition, a population structure and kinship workflow is also available in CartograPlant, which is interesting by itself because it allows to answer population genomic questions, but also because it is necessary to account for population structure and kinship for association mapping and landscape genomics analysis, which are other 2 workflows available in CartograPlant, in order to avoid spurious associations. This association mapping and landscape genomics workflows allows to identify candidate genes of resilience to abiotic and abiotic stresses, and also locally adapted SNPs. And finally, with the summary statistics of these two workflows, it is possible to perform meta-analysis, which is also possible thanks to the metadata collection and also the SNP remapping workflow. So as a conclusion, CartograPlant is intended to serve as a community resource for plant molecular ecology, and our future directions are mostly focused on providing more robust and flexible analytical workflows for a wide diversity of biological questions and data types. So your feedback here is very, very welcome. And also in providing a population structure visualization as a loaded layer in the worldwide map, which is very useful in order to put this population structure information into a geographical and environmental context that will help to better interpret the results. And finally, we are also planning a virtual training workshop for CartograPlant, and also a context for data submission via TPPS. The date for the workshop is August 22, 2022. So if you are interested in these events, please stay up to date in our websites and social media. And finally, we'd like to thank all the members of the project belonging to 4 different universities. Without their hard work and talent, this project wouldn't be possible. Also, the funding agency of this project, and of course, to you all for your attention.